You are once again welcome to the course Bio 208 Population Genetics and Evolution Unit 1, Session 2, Basic Premises of Population Genetics. And the objectives are explain the premises of the theory of microevolutionary change. Then you relate the understanding of the theory of microevolutionary change to a variety of populations. So microevolutionary mechanisms work upon genetic variability, so it is not surprising that fundamental premises that underlie population genetic theory and practice all deal with various properties of diribonucleic acid, DNA, the molecule that encodes genetic information in most organisms. A few organisms are ribonucleic acid, that's RNA, as their genetic material, and the same properties apply to RNA in those areas. So indeed, the theory of microevolution change stems from just three premises. DNA can replicate, DNA can mutate and recombine, and phenotypes emerge from the interaction of DNA and environment. The implications of each of these premises will now be examined. DNA can replicate. Because DNA can replicate, a particular kind of gene, that specific set of nucleotides, can be passed on from one generation to the next and can also come to exist as multiple copies in different individuals. Genes, therefore, have an existence in time and space that transcends the individuals that temporarily bear them. The biological existence of genes over space and time is the physical basis of evolution. The physical manifestation of a gene's continuity over time and through space is a reproducing population of individuals. Individuals have no continuity over space or time. Individuals are unique events that live and then die and cannot evolve. But the genes that are individual are potentially immortal through DNA replication. For this potential to be realized, the individuals must reproduce. Therefore, to observe evolution, it is essential to study a population of reproducing individuals. A reproducing population does have continuity over time, as one generation of individuals is replaced by the next. A reproducing population generally consists of many individuals, and these individuals collectively have a distribution over space. Hence, a reproducing population has continuity over time and space and constitutes the physical reality of a gene's continuity over time and space. Evolution is therefore possible only at the level of a reproducing population and not at the level of individuals contained within the population. The focus of population genetics must be upon reproducing populations to study microevolution. However, the exact meaning of what is meant by a population is not fixed, but rather can vary depending upon the questions being addressed. The population can be a local breeding group of individuals found in close geographic proximity, or it could be a collection of local breeding groups distributed over a land space. Evolution requires change, and change can only occur when alternative exists. So if DNA replication were always 100% accurate, there could be no evolution. A necessary prerequisite for evolution is genetic diversity. The ultimate source of this genetic diversity is mutation. There are many forms of mutation, such as single nucleotide substitutions, insertions, deletions, transpositions, duplications, and so on. For now, our only concern is that these mutational processes create diversity in the population of genes present in a gene pool because of mutation. Alternative copies of the same homologous region of DNA in a gene pool will show different states. Mutation occurs at the molecular level. Although many environmental agents can influence the rate and the type of mutation, one of the central tenets of Darwinian evolution is that mutations are random with respect to the needs of the organism in coping with its environment. There have been many experiments addressing this tenet, but one of the more elegant and convincing is replica plating, first used by Joshua and Esther Ledbeck in 1952. Replica plating and other experiments provide empirical proof that mutation occurring on DNA at the molecular level is not being directed to produce a particular phenotypic consequence at the level of an individual interacting with its environment. Therefore, we will regard mutations as being random with respect to the organism's needs in coping with its environment. Mutation creates allelic diversity. Alleles are simply alternative forms of a gene. In some cases, genetic surveys focus on a region of DNA that may not be a gene in a classical sense. It may be a DNA region much more larger or smaller than a gene or a non-coding region. We will use the term haplotype to refer to an alternative form, that specific nucleotide sequence among the homologous copies of a defined DNA region, whether a gene or not. The allelic or haplotypic diversity created by mutation can be greatly amplified by the genetic mechanisms of recombination and diploidy. In much of genetic recombination refers to meiotic crossing over, but we will use the term recombination in a broader sense as any genetic mechanism that can create new combinations of alleles or haplotypes. This definition of recombination encompasses the meiotic events of both independent assortment and crossing over, and also includes gene conversion and any non-meiotic events that create gene combination, that's new gene combination that can be passed on through a gamete to the next generation. Cellular reproduction and diploidy can also be thought of as mechanisms that create new combinations of genes. The distinction between mutation and recombination is often blurred because recombination can occur within a gene and thereby create new alleles or haplotypes. The phenotype is a measurable trait of an individual, or as we see later, it can be generalized to other units of biological organization. Previously, genes can only be identified through their phenotypic effects. The gene was often named for its phenotypic effect in a highly inbred laboratory strain maintained under controlled environmental conditions. This method of identifying genes led to a simple-minded equation of genes with phenotypes that still plague us today. Almost daily, one reads about the gene for coronary artery disease, the gene for trail seeking, and so on, equating genes with phenotypes. That is reinforced by metaphors appearing in many textbooks and science museums to the effect that DNA is the blueprint of life. However, DNA is not the blueprint for anything. That is not how genetic information is encoded or processed. For example, the human brain contains about 10 neurons and 10 neuronal connections. Does the DNA provide a blueprint for these 1015 connections? The answer is an obvious no. There are only about 3 billion base pairs in the human genome. Even if every base pair coded for a bit of information, there is insufficient information storage capacity in the human genome by several orders of magnitude to provide a blueprint for the neuronal connections of the human brain. DNA does not provide phenotypic blueprint. Instead, the DNA information encoded in DNA controls dynamic processes such as axonal growth patterns and signal responses that always occur in an environmental context. There is no doubt that environmental influences have an impact on the number and pattern of neuronal connections that develop in mammalian brains in general. It is this interaction of genetic information with environmental variables through developmental processes that yield phenotypes, such as the precise pattern of neuronal connections of a person's brain. Genes should never be equated to phenotypes. Phenotypes emerge from genetically influenced dynamic processes whose outcome depends upon environmental context. So in review, microevolutionary mechanisms work on the principles that genetic variability The fundamental premises that underlie population genetic theory and practice focus on the properties of deoxyribonucleic acid, the molecule that encodes genetic information in most organisms. Microevolutionary change hinges on the three premises that these include the fact that DNA can replicate, DNA can mutate and recombine. Phenotypes emerge from the interaction of DNA and the environment. The existence of genes over time in space is the physical basis of evolution. The physical manifestation of a gene's continuity over time through space is a reproducing population of individuals. The ultimate source of this genetic diversity is mutation. There are many forms of mutation, such as single nucleotide substitutions, insertion, deletions, and transpositions, duplications. Because of mutation, alternative copies of the same homologous region of DNA in a gene pool will show different states. A phenotype is a measurable trait of an individual. Phenotypes emerge from genetically influenced dynamic processes whose outcome depends upon environmental context.